Welcome to this video. In this video, what we're going to discuss is the pathogenesis of atherosclerosis. Okay, so the first thing to say is that this video will be split into several different parts, and all of the parts will be placed in the playlist on the cardiovascular system. So if you're struggling to find the next part in the video, go to the playlist on the cardiovascular system and you'll find them all in order there. Okay, so let's begin with the definition of atherosclerosis. So atherosclerosis refers to the disease process in which atherosclerotic plaques, which are also known as atheromas, develop in the walls of the arteries of your circulatory system. Okay, so let's write down these key terms, which are atherosclerotic plaque and atheroma. Okay, so these are the key things that are going to actually be forming an atherosclerosis. Okay, so atherosclerotic plaque, and then another name for it is atheroma. Now, atherosclerotic plaque is the more common name to see or hear people using. However, you will occasionally hear people calling them atheromas, and therefore you should be aware that that means the same thing. Okay, so what is an atherosclerotic plaque? Well, of course, that's the question that we're going to spend the entire video answering, but simply speaking, an atherosclerotic plaque is a non-cancerous mass that forms in the wall of an artery and which will push into the lumen of the artery or extend into the lumen of the artery, and this will therefore reduce the diameter of the lumen of the artery and obstruct blood flow through that artery. Okay, so I'll repeat that. An atherosclerotic plaque, also called an atheroma, is going to be this mass that is not cancerous. So this isn't a cancerous process at all that's occurring here. It's a mass that is going to form in the wall of an artery and which will extend, encroach into the lumen of the artery and reduce the diameter of the lumen of the artery and therefore impede blood flow. Okay, so what then are we going to discuss in this video? Well, we are going to discuss the pathogenesis of atherosclerosis. How do these atherosclerotic plaques actually form? And of course, by answering that, by looking at that process in detail, we will understand the structure of these in much more detail. So I've given you the broad, simple answer as to what an atherosclerotic plaque is. It's this mass in the wall of an artery. However, you want to know more about it. What's the structure of this mass? We will see that in the video. So we are going to look at the development of atherosclerotic plaques. Where do they come from? How do they form? Okay, what we are not going to talk about in a huge amount of detail is the consequences of them forming. So of course, the consequences are extremely severe, which is why this is such an important disease process to know about. So if atherosclerotic plaques form in your coronary arteries, that can lead to ischemic heart disease, the most severe consequence of which is myocardial infarction. If atherosclerotic plaques form within your cerebral arteries, then that can lead to stroke. And if atherosclerotic plaques form in your peripheral arteries, then that can lead to claudication and peripheral vascular disease. So there are a lot of extremely severe consequences of atherosclerosis. We're not going to go through those consequences in a huge amount of detail. We're just going to be aware of them and use that to motivate our discussion of the pathogenesis of atherosclerosis. We're aware that there are very severe consequences and therefore it's worthwhile understanding where atherosclerotic plaques come from, but we're going to save the in-depth discussion of the consequences of atherosclerosis for other videos that, when I've made them, will be put in the playlist on the cardiovascular system. Okay, so we are going to focus purely on the pathogenesis of atherosclerosis. Where do these structures, atherosclerotic plaques, actually come from? And we're also going to talk about anti-atherosclerotic drugs, so drugs that you can take to try and prevent atherosclerosis from occurring and try and reduce the rate at which it's occurring. Now, if you go to pharmacology textbooks, you can find a great number of different categories of drugs that can be used to prevent atherosclerosis. A lot of them are no longer actually clinically used, so things like the fibrates, they can still be clinically prescribed as anti-atherosclerosis medications. However, they're very rarely used. So we're not going to go through all of the historical drug categories that were once used to treat atherosclerosis. Instead, I'm just going to focus on the two main categories of drugs which are now currently used to treat atherosclerosis. And the first and main one is, of course, the statins. So we'll discuss the mechanism of action of the statins in a lot of detail. The second class of drugs, the second drug, it's a single drug in its own, is the drug ezetimibe. It is still used clinically to prevent atherosclerosis, and we'll discuss its mechanism as well, what at least is understood of its mechanism. Okay, so that's the outline then for this video. We're going to discuss pathogenesis and then drugs to try and slow down the rate at which atherosclerotic plaques are produced, but we're not going to go into detail of the consequences of atherosclerosis. So if you're looking for a video on the consequences of atherosclerosis, this isn't it. Okay, so let's begin then with the actual pathological process of atherosclerosis. Now, in order to discuss the pathological process of atherosclerosis, we need to have in our heads, fresh in our heads, the structure of an artery wall. So what I want to begin this video with is a recap, revision hopefully, of the structure of a normal healthy artery wall, because it's going to be necessary for us to know the different layers of the artery wall in order to understand where atherosclerosis is actually going to be occurring. So for instance, we'd like to know where these atherosclerotic plaques are actually going to form, in which layer of the artery blood vessel are they actually going to form, so we do need to know what the different layers of the artery vessel wall actually are. Okay, so that's where we'll begin then, with a revision of the structure of an artery. Okay, so what I'm going to do is I'm going to draw a classical cross-section then of an artery, and you can imagine that this is one of, your favourite example of an artery. So just pick your favourite example of an artery. We could be drawing that. So we could be drawing the aorta, we could be drawing the carotid artery, we're drawing a big artery of the body. Okay, so I will start from the inside and work outwards, because if you start from the outside and work inwards, there's a risk that you draw the initial circle too small and then the inner structures end up completely overcrowded. Okay, so we'll start on the inside then. So the innermost layer then of an artery, test yourself, what is the innermost layer of an artery? So what is it that actually borders the lumen? Well, the cells which line the lumen of a blood vessel are, of course, the endothelial cells. So I will draw some endothelial cells here. These are the cells which the contents of the blood, so all the blood cells, the red blood cells, the white blood cells, the platelets, and all of the proteins within the blood plasma, all of these are in contact with the apical surface of the endothelial cells, which are shown here. Now, the endothelial cells are attached to a basement membrane, which is a layer of proteins, principally collagen proteins, extracellular matrix proteins, of which the main example is collagen. Another notable example of a protein that is within basement membranes that you should be aware of is laminin proteins. Okay, but what you should imagine that this is, and on my picture I've just shown it as a green line here, but what you should imagine this as is a spider's web-like mesh, very strong and solid, of proteins, and the endothelial cells are going to be attached onto this, and this is what stops the endothelial cells, detaching and whizzing off down the lumen. Okay, they are attached to this rigid basement membrane in this way. Okay, so let's label up the structures that we've got so far. So firstly, endothelial cells, and throughout the video I'll be abbreviating endothelial cells down to ECs, but I suppose I should write out their full name at least once. So this stands for endothelial cells, and then in green here we've got the basement membrane, and throughout the video I'll be abbreviating basement membrane down to BM, but again, at least once I should write it out in full. So this is the basement membrane. Okay, right, so I'll just colour code these structures in. So we'll have the basement membrane coloured in in green here, and we haven't gone for a colour for endothelial cells without underlining them in blue. Okay, right, so what's the next layer? Let's go outer layer. So underneath the basement membrane in the wall of the artery, what do you actually have? Well, you have a layer of connective tissue known as the subendothelial space, and this is going to be extremely important, so I'll write out its name firstly. So we're now going to have a layer of connective tissue known as the subendothelial connective tissue, if you're being absolutely proper, but we'll just call it the subendothelial space. So this is loads of connective tissue. It's effectively like an extension of the basement membrane. It's again majorly consisting of collagen. Okay, so it's going to be a layer of connective tissue, and of course in that layer of connective tissue you're going to have, for instance, dotted around the cells which actually make that extracellular matrix, which will be fibroblasts. Okay, oops, excuse me. So you've got underneath the basement membrane a layer of connective tissue known as the subendothelial space. Now it's quite difficult to actually draw this, so what I'm firstly going to do is draw out the thing that demarcates the edge of the subendothelial space, so the boundary of the subendothelial space. And the end of the subendothelial space is lined by a structure known as the internal elastic lamina. So I'll draw this firstly, and I'll draw this in blue. Oh, and by the way, I didn't actually tell you the reason the subendothelial space is so important for us to know about. The reason it's so important for us to know about is it's the layer in which atherosclerotic plaques are actually going to form. So these things that we are spending the entire video discussing, they are going to form in this space here, the subendothelial space, this space underneath the endothelium of the arterial wall. Okay, right, so what have I just drawn on this blue ring? This is a ring of very elastic connective tissue, so elastin, that surrounds and demarcates the edge of the subendothelial space, and this is known as the internal elastic lamina. So very elastic connective tissue, or another ring of connective tissue, but this time it's extremely elastic connective tissue. So this is known as the internal elastic lamina. And we'll see also that there is an external elastic lamina in the structure of a blood vessel wall, specifically the structure of an arterial blood vessel wall. Okay, so we will see the external elastic lamina as well. Lamina, by the way, just means layer, so it's the internal elastic layer. Okay, so why have I put the internal elastic lamina on before showing the subendothelial space? Well, it's because now it's easier to show the subendothelial space, because the subendothelial space is just all of this structure in between the basement membrane and the internal elastic lamina. So all of this is just going to be connective tissue with fibroblasts within it, and that's the layer in which an atherosclerotic plaque is actually going to form. So of course, initially I'm just drawing a lovely healthy blood vessel, so there's no atherosclerotic plaque occurring here yet, but we will see the atherosclerotic plaque forming in this subendothelial space when we come on to discuss the actual pathogenesis. Okay, now, there is a combined name for all of the layers of the blood vessel wall that we have discussed so far. So the endothelial cells sitting on their basement membrane with the subendothelial space and the internal elastic lamina, there is an umbrella term which includes all of those different layers, and this is known as tunica intima. So all of these different layers, all four of these different layers, together they are all put under the umbrella term tunica, which again just means layer. So tunica, layer, and then intima, the layer that is close to the lumen. Okay, so intima, you know, it's Latin, so it's the same sort of thing as intimate, which means close. Okay, so tunica intima, the layer that is close to the lumen of the blood vessel. Okay, and I probably should just point out that this, I hoped this was obvious, but I will point it out explicitly. That, of course, is the lumen of the blood vessel where the actual blood is moving. Okay, right. Next layer then. The next layer is going to be tunica media. which is the layer of smooth muscle cells of the artery wall, and also it's going to contain a lot of elastic tissue. Now, the balance between smooth muscle cells and elastic tissue varies depending on which artery you are actually looking at. So if you're looking at an artery like the aorta, which is called an elastic artery, this next layer outside of the internal elastic lamina is going to consist of mainly elastic tissue and then a few smooth muscle cells. Okay, and that's because the aorta needs to be able to expand when blood is pumped into it by the heart during systole, and then retract back down during diastole. Whereas, if we're talking about a muscular artery, so for instance the carotid arteries, they're very muscular arteries, their next layer, the tunica media, is going to consist a lot of smooth muscle cells and much less elastic tissue. Okay, so the next layer consists of smooth muscle cells and elastic tissue. It's known as tunica media, and the balance between smooth muscle cells and elastic tissue will vary depending on which artery of the body you're talking about. If you're talking about an elastic artery, such as the aorta, that's the principal example, then it will have a huge amount of elastic tissue. If you're talking about a muscular artery, again, the principal example is the carotid arteries, then it will have much more smooth muscle. Okay, right, so I'll draw some of these smooth muscle cells. I'm not going to draw all of them, but let's say here is a smooth muscle cell, so they have this characteristic spindle shape, and you're going to have circles, effectively, rings of smooth muscle cells surrounding surrounding the artery. Okay, and the idea here is that the length of the smooth muscle cell is oriented going around the artery. So now, when these smooth muscle cells contract down, you can imagine that what's going to happen is it's going to reduce its length, and I'm just going to colour them in red so that they look more like muscle cells. Okay, so here are our smooth muscle cells. So you can imagine when they actually contract, they're going to reduce their length. And if you imagine having an entire ring of these things and they're all reducing their length, then what's that overall going to do to the circumference of the ring of smooth muscle cells? Well, of course, it's going to get smaller. And when the circumference of a ring gets smaller, I'll just make sure this is visible, when the circumference of a ring gets smaller, the diameter of that ring gets smaller, so that's going to constrict down the artery. So the smooth muscle cells are going to determine the diameter of the lumen of the artery. Okay, so again, it's quite difficult to actually show this layer, so instead what I'll draw on first is the boundary of it, and the boundary of it is going to be the external elastic lamina. So I'm going to draw on another elastic lamina here in blue, like so, and this is the external elastic lamina this time, and then all of that space in between the internal and external elastic lamina, I'll colour this in red, like so, and this is all going to be tunica media here. And as I say, it will consist of smooth muscle cells and elastic tissue, and the balance between those will vary depending on which artery you're looking at. Okay, so all of this layer here, from the internal elastic lamina here to the beginning of the external elastic lamina, which I'll label on in just a moment, this is now called the tunica media of the artery wall. So tunica means, again, layer, media means middle, so this is the middle layer of the artery wall. Okay, right, and the tunica media, it controls the diameter of the blood vessel, because the smooth muscle cells, they can vary their contracted state, and depending on how contracted they are, that will determine the diameter of the artery. Okay, now, the next layer then, we've already got it drawn on, but I'll label it up. So this now is the external layer of elastic connective tissue, known as the external elastic lamina, and it demarcates the edge of the tunica media. Okay, so that's the external elastic lamina, and then finally we've got one more layer surrounding the external elastic lamina, and this is going to be full of connective tissue. Okay, so I'll show this like so, in yellow here. So there is a layer surrounding the external elastic lamina, and this is now known as the tunica adventitia, and this is connective tissue, so it's collagenous connective tissue mainly again, and this will connect the artery now to the surrounding structures. Okay, so this final layer then, this is known as tunica adventitia, or it's also called tunica externa. So layer adventitia. I don't know what adventitia stands for. Externa is easier. That obviously means the external layer. Okay, I would imagine that adventitia means something along the lines of external as well, but I don't know the origin of that word. Okay, so the major layers then of an artery wall are tunica intima, which consists of the endothelial cells, the basement membrane, the subendothelial space, and the internal elastic lamina. Then tunica media, which consists of these smooth muscle cells and elastic tissue. Then you've got the external elastic lamina in between tunica media and the final layer, which is tunica adventitia, which is a connective tissue layer that will attach the artery to surrounding tissues of the body. Okay, so there is our picture then of a normal artery structure. So I hope we're now all happy with the different layers of the artery wall. In particular, I hope you're happy with where the subendothelial connective tissue or the subendothelial space is, because that's the layer in which atherosclerosis is going to occur. So if you can imagine putting a huge great mass in here, you can imagine what it will do is it will bend this basement membrane outwards, and the endothelial cells will also go outwards, so they'll extend now into the lumen because there's a great big mass sitting in the subendothelial space, and that's the principle of what is going to happen in atherosclerosis, in the formation of an atherosclerotic plaque. So we'll have a break here, and in the next video what we'll do is actually begin the pathogenesis process.